So today we are going to know about the lytic and lysogenic cycle. Lytic and lysogenic cycle are two different reproductive cycles of bacteriophage, the virus that infect the bacteria. These cycles describe how virus replicate inside the host cell. So lytic cycle is active infection and destruction, where the lytic cycle is a virulent cycle where the virus immediately hijacks the host cell machinery to make many copies of itself and destroy the host cell in this process. So first the lytic cycle, the attachment process is done. The phage attaches to the specific receptor on the bacterial cell wall. Then penetration happens. The viral DNA or RNA is injected in the bacterial cytoplasm. Then the biosynthesis takes place. The host cell's machinery is hijacked to synthesize a viral component, that is the DNA and protein. Then assembly happens. A new viral particle, that is a virion, are assembled inside the host. Then lysis, that is the host cell burst open. Lysis happen, releasing newly formed virus that can infect other cell. Example are the T4 bacteriophage and infecting the E. coli. So prophage is a bacteriophage. It is often shortened to phage and genome that is integrated in the cellular bacterial chromosome or exist in the extrachromosomal plasmid within the bacterial cell. This integration of prophage into the bacterial host is a characteristic step of the lysogenic cycle of a temperate phage. The prophage remain latent in the genome through multiple cell division until activated by the external factors such as the UV light, leading to the production of new phage particle that with lysis the cell and spread. As ubiquitous mobile genetic element, prophage play important role in the bacterial genetic and evolution such as the virulent factors. Upon detecting of the host cell damage by the UV light, certain chemicals, the prophage is excised from the bacterial chromosome in the process called prophage induction. After induction, the viral replication begins via the lytic cycle. In the lytic cycle, the virus commands the cell reproductive machinery. The cell may fill with new virus until it lyses or bursts. So it may release the new virus one at a time in a exocytotic process. So the period from the infection to lysis is termed as a latent period. A virus following a lytic cycle is called virulent virus. Prophage are important agent for horizontal gene transfer and are considered part of the mobiloma. So genes are transferred by transduction as a prophage genome is imperfectly accessed from the host chromosome and integrated into a new host, that is a specialized transduction, or a fragment of host DNA are packed into the phage particle and introduced in the new host. Generally transduction. All families of bacterial virus have circular, single-stranded or double-stranded DNA genome or replicate the genome through the rolling circle replication. Have a very temperate membrane. During infection by the bacterial pathogen, spontaneous prophage release from the bacterial chromosome is common, and the presence of the deoxycholic acid in the intestinal environment can promote induction. For example, clostridioides difficile, that is C. difficile biofilm formation as well as the induction of prophage release. After that, zygotic induction occurs. Zygotic induction, when a bacteria carry the DNA of bacterial virus, transfer into the own along with the viral DNA prophage in the new host cell. So this has an effect of causing the host cell to break apart. DNA of the bacterial cell is silenced before entering the new cell by a repressor protein which encode by the prophage. So upon transfer of the bacterial cell DNA in the host cell, the repressor protein is no longer encoded for and the bacterial cell's original DNA is then turned on in the host cell. So this mechanism eventually lead to release of the virus as a host cell. Splitting open the viral DNA is able to spread. So bacterial conjugation and the contribution to the earlier repressive model of gene regulation, which provide an explanation of how the lacoperon and lambda bacteriophage genes are negatively regulated. Now, bacterial lambda is able to undergo a type of recombinant repair called prophage reactivation. So, prophage reactivation can occur by recombinant between a UV damage infection phage, that is lambda chromosome, and a homologous phase genome integrated in the bacterial DNA and existing in the prophage state. So, prophage reactivation in the case of phage lambda appeared to be an accurate recombinant repair process that is mediated by the REC-A and red plus gene protein. These are the steps of biosynthesis. Then assembly happens, that is the maturation. The viral DNA and protein assemble here into the new virion. Then release happens. The host cell bursts open. Hundreds of new phages are released. And the result is that the cell dies and the new virus is spread. Then lysogenic cycle. In this, the viral integrated in the host genome. No immediate destruction is seen, and it is temperate phage. That is, lambda phage is an example of lysogenic cycle. First step is attachment and entry. First it will attach, it will enter. Then integration happens, the viral DNA integrate into the bacterial chromosome. It becomes the inactive virus or prophage. And after that, this silence the replication. Every time the bacterium divides, it copies the prophage too. It will copy it. Then induction happen due to stress like UV light, chemical. Prophage may wake up. It enters the lytic cycle and kill the host. Here the virus hides and it multiply passively without harming the host until triggered. So lytic versus lysogenic, if we compare, so the features we have seen, it is a type of virulent phage, lytic cycle, whereas lysogenic cycle is a temperate type of phage. The host cell fate is destroyed in lytic cycle, whereas it is survived initially in a lysogenic cycle. The viral genome fate in lytic cycle is replicate independently, whereas in lysogenic cycle it is integrated into the host genome. The symptoms of lytic cycle are acute, whereas in lysogenic cycle it are latent or dormant. The example of lytic cycle is T4 phage, and in lysogenic cycle it is lambda phage. So the lysogenic phase can become lytic when host is under stress, DNA damage occurs, or it ensure the viral escape before the host die naturally. In that time, the lysogenic cycle can become lytic cycle. For example, real life relevance if you want, HIV or herpes virus. They show similar latent behavior in human. Understanding this cycle help in the developing antiviral drug, gene therapy and biotechnology tools. Now let's know about the virus throat interaction as a phage cycle as a part of immune evasion and microbial ecology. Phage evolve attachment specifically to the bacterial surface receptor. The immune system can recognize lytic phage in a bacteria indirectly, aiding the adaptive immunity. Then lysogenic phase is temperate phage, often carry gene for toxins or antibiotic resistance. Example, Cronibacterium diphtheria, which carry the diphtheria toxin gene from the prophage, and this is called the lysogenic conversion. The host gain new trait from the prophage. So it have viral host interaction in the immune invasion and microbial ecology. In that also it is important. Virus like bacteriophage infect bacterial cell and can follow two main strategies. We know that the lytic and lysogenic cycle. The lytic cycle is immediate replication and the host cell is destructed, whereas in lysogenic cycle the viral genome integrate into the host genome as a prophage and remain dormant. So these cycles reflect an evolutionary balance between the rapid viral speed, that is a spread, that is a lytic cycle, and long-term survival and silent spread is a lysogenic. Thanks for watching. Do like, share and subscribe.